dum. I was just checking that for you, Robertin. Is lenticuli. Which means pimple or freckle. And they're found on the surface of Europa. So if you you want to comment on you know, a nephew or niece's acne, you could say that she has a bad case of lenticuli, or he. There you go. If you want to be fancy and not offend them. Okay, so can you hear me okay today? Cool. I have to admit that I haven't actually practiced my Hal voice. I forgot, other things got in the way. Okay, so let's see. Let's do this. See, there you go. This is what lenticuli on Europa look like. They're basically thought to be evidence of water circulation underneath the surface of the ice. They're kind of upwellings. So you can see, I don't know who Professor Manga is. That's a pretty cool name. You can see those pimples here amongst the ice rafts. I think which is pretty cool. Anyway, let's okay, so first up, anybody got any questions about the Galilean moons and Io stuff? Pardon me, because I think that would be good place to start, a good way to start the class, I guess. If you do, type them up into the chat box, and I'll try and keep an eye on that as we go through. So, all right, what I really wanted to talk about today, and this is kind of a little bit of a not a tangent. It was really in response to what Paul asked about or commented on in terms of Io. Let's see, is Paul here today? No, he's not. Oh well, he's lost out. Hopefully he'll catch it another time. So yeah, so essentially his question was, you know, or statement was that Io would seem to be a pretty terrible place for life. And I was like, well, you know, it's there are things that on Earth that could probably live there quite happily, to be honest. And so really wanted to talk about radiation resistance, and partly as well because I love that picture. Can you all see that? It's my favorite Arnold Schwarzenegger picture, of which obviously there are many. And it's just so I can say the phrase Conan the Bacterium. Anyway, so it's kind of an interesting topic because you don't really think of ability to resist radiation as being in like an extreme environment, right? It's not like high temperatures or high pressure or high salinity, things like that, but it is very much the same context, and that's because these radiation, especially ionizing radiation, is extremely bad for you, right, as an organism. And so essentially there's a bunch of different types of radiation, and you can broadly group those either into ionizing and non-ionizing radiation, or particulate and electromagnetic radiation. It kind of depends on I don't know what you ate for breakfast that morning or your particular preferences. And so ionizing radiation is the most damaging. As you would expect by the name, it produces ions, so it has sufficient energy, either through mass or its wavelength, to knock electrons off of atoms, which is all around 100% bad for you, because those electrons then go on to do all kinds of mischief in themselves. So like they're free radicals, essentially, superoxides. And also the things being turned into ions generally isn't good for their structure, right? So it has a big effect on protein structure and function, as well as stability of DNA, its ability to maintain its structure. And so ionizing radiation, I've never really understood exactly what neutral particles do, so we're not going to talk about those. But alpha, beta, and gamma radiation are the three types of ionizing radiation that I'm kind of more familiar with. And essentially it goes from large, chunky, and slow, which is alpha, right? So that's 2 neutrons and 2 protons. A good example of that is radon, right? So if you have live in an old stone house and you go down into the basement, those rocks kind of produce naturally alpha particles. And generally speaking, they're not very bad for you because they don't penetrate very far. I think basically a sheet of paper is enough. But if they get inside you, they can do a enormous amount of damage, right? So their, I don't know, innocuousity is more based on the fact that they don't penetrate into your body. Beta particles are electrons, and so they penetrate a little bit further. They cause actually most predominantly produced in nuclear explosions, which you want to try and avoid anyway at the best of times. And so these cause, they also don't penetrate very far, further than alpha particles. And they cause what are called beta burns. Now, gamma and also X-ray is kind of like a weaker version of gamma radiation. Gamma radiation is entirely scary. And so gamma radiation is a electromagnetic wave particle. I've never really got the difference. I think it depends on how you observe them. And it is on the far, if you have blue on the left and red on the right of a electromagnetic spectrum, it's way over to the left, right? So it's past blue, past UV, past X-rays, and then you have gamma rays. And it's very short wavelength, very high energy. Energy is basically inversely proportional to wavelength. So that's why the shorter wavelength UV radiation is more damaging than the long wavelength UV, generally speaking. Anyway, so that causes a lot of major problems. There's also UV, which I've talked about already a little bit. UV tends to cause DNA damage. generally speaking, called thymine dimers, which are when you have two T's adjacent to each other, they get stuck together and then they get repaired badly, which causes mutations. That's where skin cancer arises, essentially, as UV-induced mutations in genes that either control cell proliferation or um, cell death, things like that. So gamma radiation is really, really scary. That will kill you very quickly, and it generally does most of its damage by breaking DNA, but it can also really screw up proteins as well through its ionization properties. So, and we'll talk about that in a little bit. So anyway, that's usual fun clip art. Let's just check, see if nope. So obviously, it's no news to you that radiation isn't good for you, and use a couple of different ways in which you can measure how bad it is for you. You can either use absorbed dose, which is measured in grays. I don't know why. And you can use essentially the effective dose, which depends on, how what clothing you're wearing, how heavy you are, how long you've been exposed for, if it's acute versus chronic, things like that. But essentially, about 5 grays of radiation is you're dead within a couple of weeks, or about 6,000 sieverts, which you want to avoid too. So here's generally a quick overview, a little bit of a joke. I don't know if you can get that. Anyway, three main problems that radiation causes, depending on the types of radiation. As I said before, UV causes these thymine dimers, which you can see here, which then get fixed poorly. The DNA repair mechanism that fixes those is error prone for some reason. I don't know why. Ionizing radiation typically generates either free electrons or hydroxyl ions, right? Either which are generally pretty bad for you. And gamma radiation causes double-stranded breaks. So another reason why gamma radiation is so scary is because of its short wavelength. Essentially, if it's kind of doing this, the potential for it to interact with something as it passes through your body is a lot higher than a longer wavelength radiation, which is, I don't know, more likely to miss you, let's say. And so gamma radiation is extraordinarily high energy, and it's very, very bad for you. So there are organisms that can live perfectly happily on high radiation, though, and the leading candidate, I don't think necessarily because it's super extra better than others, but mostly because it was discovered first, is a bacterium. Well, actually, it's an archaeon. I think I'd have to double check that. Called Deinococcus radiodurans. No, actually, this is a bacterium. Another one I'm thinking of is an archaeon. So we die after about 5 grays of radiation, even Conan. I'm not sure whether Arnie would be quite so tough now, he's a little bit older. Whereas D radiodurans can survive and grow, right? This isn't just tolerate, this is kind of shrug it off like, meh, whatever, on about 5,000. And it can actually tolerate with quite a few deaths, about a third of those cells would survive on 15,000 gray. Right, so it is astonishingly resistant. Angela, you don't have to raise your hand. It's kind of cute though. Just unmute your audio and ask a question. Angela? Oh, grays. Oh, that's okay. Don't worry. Exactly. So they can tolerate over 1,000 times the radiation that will kill us. That's how astonishing those things are. And there are several other bacteria and archaea that can do the same too. Right, so this isn't the only one. It was just the one that was discovered first. And what was it? I can't remember the exact context, but I think there was someone was um, trying to sterilize food, bread, or I don't know, baked beans, something or other, with um, radiation. And they actually found that even though they bombarded it with these crazy amounts of radiation, it the food still spoiled. And the reason for that was because of these bacteria. Hang on, sorry, I'm trying to get chat back. Where the hell are you, chat? There you go. Okay, cool. So, and the funny thing is as well is that, so one of the big questions, if you think about it in evolutionary terms, right? So typically you have a behavior or an attribute or a trait which confers survival or fitness in a particular condition, right? And so obviously radiation resistance allows these things to grow on gonzo amounts of radiation. But the question in evolutionary terms is, well, where did that radiation come from? We don't have any radiation sources on Earth that are that high that are naturally occurring, right? Even uranium deposits in Utah and things like that don't hit those levels, right? Because otherwise the people who mined it would be dead before they even picked up a shovel. Um, and so they actually think that the radiation resistance is tied into desiccation resistance. So the ability of a bacteria to resist drying out, like losing almost all of its water and then recover again, seems to be what gives its ability to resist radiation. It's like kind of a 2-for-1 deal, I guess. Because if you take away its other things which allow it to resist desiccation, pardon me, it's much, much less able to survive and grow on high radiation levels. So that's kind of an interesting thing because, again, desiccation or low water availability is another example of an extreme environment, right? Which we talked about. What was we talking about with that? I know we're talking about lichens. Yeah. Anyway, I like lichens. So essentially, there's a bunch of different things that go on, right, in terms of radiation, and one of them is the effect on proteins and lipids, which we'll talk a little bit about later on. But the big one is fixing double-stranded breaks, right? So if you or I are exposed to radiation, and that ionizing radiation is especially such as like gamma radiation, if that interacted with the DNA in our cell, most likely it would cause it to break, right? And it would just separate the both strands of DNA, so you have two ends kind of flapping in the breeze, basically. So in cells which are not growing, typically, they're in stationary phase, or there isn't another copy of that chromosome around, like if you had a sister chromatid present, then your cells would try and just stick those ends back together, right? So if you have one break, it's like, well, we've got two ends, you just stick them back together, like what ligase is doing here, to just making ends meet. However, if you have more than one DSB, how do you know which end goes with which? Because there's nothing inherent in the end of a bit of DNA. It's just either a phosphate or a sugar, right? There's nothing to say this one goes with this one. So what happens then is we have this repair mechanism called non-homologous end joining, NHEJ, and essentially the enzymes just go free end, stick them together. Now this free end could be from chromosome 2, this free end could be from chromosome 5, doesn't matter, they just get stuck together. Because if you leave them hanging around, then that's even worse than not fixing them, even if you're going to fix them badly. So fixing double-stranded breaks can be extremely error-prone and cause all kinds of issues, such as dislocation of chromosomes. You could even lose bits of chromosomes, right? If you have multiple breaks within the same chromosome. And there's actually a phenomena called, here you go, here's another word of the day. Oh, hang on, I missed a whole bunch of chat. Where who's the dumb person to go into these nuclear reactors and scrape samples of? So these weren't isolated from nuclear reactors. They were isolated naturally available, and you'll tend to find them in all kinds of what we would consider unpleasant places, like sewerage, kind of peat bogs, things like that. You even find them in the soil, right? So these bacteria aren't. Here we go. These bacteria aren't extreme bacteria in the sense of, say, Thermophilus aquaticus or something like that. Something that lives in high temperature. It's a mesophile that lives in normal temperatures, normal environments. Yeah. But to Sam, that's kind of what those people in Chernobyl were told to do. They were told to just go in and fix the mess, right? And that's why we have where is it? These data up here. So if you look at the top of that graph, it says typical dosage recorded in those Chernobyl workers who died within a month. So that's how we know how much, how many sieverts are enough to kill someone, because a bunch of those people were sent into the Chernobyl reactor to clean up the mess. Actually, on a kind of side tangent, I think one of my daughters did a science project on Chernobyl last year, I think. There's you probably all know of the elephant's foot, which is basically all the material of the reactor which melted together and then kind of slumped down underneath the where the reactor was. You can't actually take pictures of that, not directly, because the radiation is so intense it will destroy the camera or overexpose the film. You can only take it using like a series of mirrors. So radiation is not something to be trifled with. It's a very, very scary thing. Anyway, getting back on track. So a much better way of fixing double-stranded breaks is to use another copy of that chromosome as a scaffold. So essentially, if you have an intact copy of a chromosome and then you have a free end up here, you can now go, okay, where does this match, right? And so you're able to line that broken end up with that copy. And then you take one bit of that broken end and you kind of patch it into that existing chromosome, which is what you can see at the bottom there. And so in doing so, you fix that break without introducing any errors. And that's homologous recombination, right? Which you're probably familiar with from meiosis. And so that is absolutely awesome. That's like the best way of doing things. But you can't do that unless you have an exact copy of that chromosome present. Homologous chromosome isn't sufficient. And so you can only really do that during cell division when you have those sister chromatids around. So typically we're unable to do that if we have DNA damage like that. However, D radiodurans, this thing up here, and they grow in these like, they always look to me like, you know, when you get oven buns. I don't know if you ever, you know, you get the little frozen ones, you stick them in the oven and you get buns come out after a little while. Hence the phrase bun in the oven. They form this, these kind of tetrads, typically. They have a funky way of dividing. I don't believe that they share anything between them, right? They don't share DNA, but they're, yeah, there you go. Sweet rolls. That'd be nice. Mind one of those right now. Anyway, sorry, I get distracted by food really, really easily. So one of the tricks that they use is they actually have, depending on conditions, about 4 to 10 copies of every gene. And so you could see it here. They've got these essentially multiple chromosomes. They only have 2 chromosomes, which is kind of odd anyway for a bacterium. But they have multiple copies of each of those genes scattered throughout those 2 chromosomes. So whenever they receive a serious dose of radiation, they simply fix it really quickly via homologous recombination. And they're very, very good at doing that. The funny thing is their DNA isn't more resistant to damage. It breaks just as badly as ours does. They just have a lot better way of fixing those breaks, right? So they're able to use those, those copies which are always around to fix them. And I think for a human cell, like 200 double-stranded breaks is lethal to the cell, and these are quite able to deal with 1,000 or more. Okay. The other thing which is really beneficial for them is that, so we talked about earlier a little bit about how proteins get damaged by radiation, and they tend to get damaged because essentially ions are created in the side chains of amino acids in their structure, and that affects how the structure folds, how it stays together, right, which is equal to function in proteins. And they also have, a lot of them, enzymes particularly, active sites, which quite frequently have a ion as a cofactor. So if you start introducing other ions either in that structure or that can go into that structure, those proteins don't work very well. And if you, hang on, get off of there. The proteins that fix, the things that fix DNA are proteins, right? So essentially radiation causes a double whammy where not only does it break your DNA and potentially introduce mutations, it also damages the things that fix those problems, right? So you're affected twice over, essentially. Not only is stuff damaged, but your ability to repair that damage is inhibited. So this is effectively worse, right? And so D-radiodurans has a very robust DNA repair mechanism, right, which is very resistant to radiation. And so kind of dotted around in here, you'll see MN, I think it's even 2+, I'm not sure. Pardon me. Which is manganese, not magnesium, manganese. And so manganese is a very potent antioxidant, right? So if you look here on the left, this is basically a survival graph, right? So here we got sugar. These are all nasty things, basically, apart from thermophilus. I'm not sure what that does. But Shigella, Neisseria, Pseudomonas, these are all pathogens, right? So these are knocked off pretty quickly by, you know, 2 gray, well, that's kilograys. So that's 2,000 grays. So that's even higher than anything that we can tolerate. And if you look at D-radiodurans all the way over here, that basically has a 10, what's that? A log -1 decrease. Is that a log? I'm not very good at math. It survives pretty well. There you go. All the way up to 16, 17,000 grays. And if you look at what's in the cells, what you'll find is that they have a lot more manganese, right? So this is the survival by dose, basically. And this is a ratio of manganese to iron. So we use a lot of iron in ions in enzymes as cofactors. Yes, it's the iron and hemoglobin, right? For example. However, basically radiation resistance correlates with how much manganese you have, right? Manganese essentially replaces a lot of the other metallic cofactors, the metal iron cofactors like iron and magnesium, and it seems to protect those active sites and make them more stable, right? So they're less likely to be disrupted by radiation. And so essentially, not only does D-radiodurans have a kind of the tools with which to fix its DNA more quickly and more effectively, but also those tools are naturally more robust and resistant to the effects of radiation. And so these things are actually thought to, remember what it's called now, Hanford. If you ever look up Hanford, Hanford, it's a site or former site of a series of nuclear reactors in Washington State. And so basically the standard approach for nuclear reactors is when you get, the uranium fuel is spent, right? It's decayed sufficiently to other things that it doesn't produce enough energy anymore. They just put them outside into ponds and just leave them there. And Hanford was on the banks of the Columbia River, right, which is a huge river in the Northwest that goes all the way from Idaho and Montana all the way out to the Pacific between Washington and Oregon. And so they found that this radioactive water was basically seeping into the Columbia. So anyway, D-radiodurans is being developed as a bioremediator. So essentially it's a bacteria that can resist high levels of radiation. So if they can put into it enzymes or proteins that can kind of, suck up that radioactive material or whatever other nasties are around in the soil at the same time, they can use it to clean up contaminated, particularly radioactively contaminated sites like Chernobyl or Fukushima in Japan, for example. So it's not just a kind of a curiosity, it's actually potentially a really useful tool as well. So maybe if we were able to get to Io and explore some of those lava tubes, maybe we'd find something that looks like that. hopefully we're not going to find something that looks like that. I would be thoroughly terrified if Conan the Barbarian kind of jumps out from around the corner on Io. Anyway, so that's pretty much it for me today. Does anybody have any questions about any of this stuff? It's a fairly short one. So the plan for the rest of the week is to look at Europa and Ganymede on Friday. So I finished the PowerPoint, but I haven't had time to do the audio walkthrough of it. So that'll go up probably this morning, actually. It'd be nice to get that out of the way and done with. And then after that, we will have a quiz on Monday, and then we'll get on to, what do we need to talk about? Halophiles after that. And then I think we're going to start on, start looking at exoplanets, basically. So yeah, that's the next week or so for us in class. Is everybody getting the material that they need and they feel like they're being able to stay on top of the stuff? Yeah, kind of. So it's obviously different because it's a lot more self-guided in terms of education. But what I would think is, if you can, just go through stuff and just make notes of or lists of things that just don't make sense, or you're unable to kind of focus on enough to really get it, or things like that. Written notes. Yeah, I think I've got, not for all of them, Connie. Some of them I do. I can post those up as well. And it is on my list of things to do to get the kind of study guide up and going. I've just been very focused on trying to get caught up and maybe a little ahead of things for the lectures. So that's another thing on my list of things to do. I'll try and get that up by the end of the week. That's another thing. So get to you in a second, Pedro. So for the others, in terms of staying focused and having guidance, so really just work through the PowerPoints ahead of time is a good idea. Listen to the audio if you can. You know, I may send you to sleep, maybe not. But then bring a list of questions, you know, the stuff that doesn't make sense or you need clarifying or whatever, with you to these sessions, because these are really just a very quick kind of overview, picking on different like key points, but they're not quite as thorough as when I go through each slide individually with the audio. So they're very complementary, right? But what we're doing today is, I think by its nature, kind of focusing on those key bits, which are, I don't know, most interesting maybe, or most relevant, but it's not as thorough a picture as the audio walkthroughs of the PowerPoints. That's why you have both, essentially. The overviews, oh yeah, I mean, it's kind of more fun, but you have all of them, right? That's the point. In terms of passing class, mostly using the PowerPoints. So the PowerPoints are the material on which the exams will be based, or the quizzes and exams will be based. Almost entirely. But you do need to listen to these sessions. There shouldn't be anything extra in these sessions that aren't in the PowerPoints, but there may be stuff which is expounded on more thoroughly. And also, when we go through these PowerPoints, either in audio or online, that will give you a sense of what are the key things that you need to get, right? not all the information in these is kind of equally weighted really in terms of importance. And so that's what these sessions are very useful for. So getting to Pedro's question about quiz and exams. So quizzes and exams will be on Blackboard. They will be multiple choice, almost certainly. I haven't ever done an exam on Blackboard, so I'll have to, it's going to be a learning curve for me. Same format as before, 10 questions for the quizzes, 30 questions for the final. For people that need accommodations, there is a way to provide extra time for that, and I just need to check in with DSS the best way to do that. So if you do have accommodations and you have things scheduled after this class, then let me know and we'll figure out a way of getting you more time. I haven't quite figured that out yet. I have that issue with my genetics class. So yeah, everybody that needs accommodations will be able to get them. That's not an issue at all. But there will be open book online exams that are timed. They're going to have the same time allowance as we had for the prior quizzes and exams. So that's going to be what, 30 minutes for quizzes, or 25 minutes for quizzes, and then the probably 55 minutes for the final exam. But yeah, you'll know what they're like when I get them done. That's a couple of more lectures need to be done, and then working out how to do exams and quizzes on Blackboard is the next thing to learn. So it may be a little bumpy, but hopefully it's going to go fairly smoothly. Cool. All right. Anybody else have any other questions? Oh, awesome. All right. So I shall see you on Friday. Keep an eye out for the announcement for the Europa and Ganymede lecture going up. Oh, hang on. Another question. Mariah, it will be in the scheduled time of class. So it will start at 9 a.m. on Monday, and it will end, for those who do not have accommodations, at 9:25 a.m. on Monday. The reason for that is you'll get to see the answers afterwards, and so obviously I can't have some people taking it before others, and also I know that there are people that have other classes or work or jobs or different requirements on their time, so I know that I'm guaranteed to get you in this time period, and so I don't really want to do it any other time other than that, because that will cause more problems than it solves. But if you are unable to take it at that particular time because of stuff that comes up, let me know, and then we'll figure out a way of doing that. Cool. All right, everybody. I'm going to go make myself a cup of coffee. If you got any questions in the meantime between now and Friday, just send me an email. I'm always going to be checking it from time to time. Okay. Have a great day. Yes, we will, Mariah, to go through the questions, unless you have accommodations, in which case you'll still be taking your quiz and you will not be able to take part in the Zoom run through for obvious reasons. Yeah. Cool. All right. Catch you later. Again, any other questions that occur to you after we finish this, just send me an email and I'll answer them as quick as I can. Cool. All right. Catch you all later.